Welcome to the Bovine Podcast, where we tackle the tough questions dairy farmers are asking, but aren't afraid to ruffle a few feathers along the way. Today, we're diving into a bombshell revelation that's rewriting the rulebook on mastitis management. For decades, we've treated E. coli mastitis like a specialized pathogen, a bacterial assassin with a hit list for udders. But new research says we've been dead wrong. What if these bacteria aren't cunning killers? Just opportunistic survivors thriving in the chaos of your barn? We're breaking down why the term pathogenic is on its way out, how bedding hygiene might be your best vaccine, and why your antibiotic protocol could be backfiring. Strap in, this isn't your granddad's mastitis talk.
All right, you're tuned into the deep dive, and today we're tackling something that could really flip the script on how we've been battling a major headache in the dairy barn. E. coli mastitis. Prepare yourselves, because it turns out everything we thought we knew might be, well, a bit off.
A bit off is, maybe putting it mildly, we've been looking at some really fascinating research, especially one study from the Journal of Dairy Science.
Okay.
And it suggests that our whole long-standing approach to E. coli mastitis might have been, well, aimed at the wrong target entirely.
Wow.
We could genuinely be talking about $1 billion misdirection in how we've tried to fight this thing.
$1 billion. That's a staggering amount of discarded milk, vet bills, everything.
It really is.
So what's this big revelation then? Is it like a secret E. coli conspiracy we didn't know about?
Not quite that dramatic, no secret lair. The real core of it is about understanding what these E. coli actually are.
Okay.
Are they specialized udder attackers, pathogens designed for the job like we've mostly believed? Or are they simply opportunistic bacteria, ones that are just really, really good at making a home in the mammary gland when they get a chance?
Okay. So the difference between like a trained invader and just an adaptable gatecrasher who sees an open door.
That's a perfect analogy. A targeted strike force versus adaptable gatecrashers.
So for ages, the idea was mammary pathogenic E. coli, MPEC, that was the term.
MPEC, exactly. These were seen as the specific bad guys, the ones with unique tools, specific virulence factors designed to, well, mess with the udder.
Like tiny bacterial ninjas with udder-seeking capabilities.
That was pretty much the prevailing wisdom, yeah. And it made a certain kind of sense, logically.
Sure.
The thinking was, if they're specialized, we can find their unique weapons, those virulence factors, and then, develop specific vaccines or treatments against them.
Target the weapons.
Exactly. The MPEC classification sort of put them in a box with other E. coli types that cause specific problems elsewhere, like in the gut or urinary tract.
But now the buzz, the new thinking, is shifting towards mastitis-associated E. coli.
MAEC,
Yeah.
Yes. And it sounds like just a small name change, doesn't it?
Yeah, seems subtle.
But the implication is huge.
Yeah.
It's fundamentally different.
How so?
Well, the MAEC idea suggests these bacteria don't necessarily have a unique set of disease-causing genes just for the udder. Instead, they seem to be more of an ecotype. Think of it as bacteria that have just evolved to be really fit, really successful in the mammary environment.
Okay.
But maybe without needing those dedicated attack genes we assumed they had. It's like being good at the local talent show versus being a world-famous opera singer. Different skill set, different context.
Gotcha. So this isn't just lab coat terminology changing. You mentioned Dr. Olson emphasized this.
Absolutely. His point was, this distinction isn't just semantics. It fundamentally changes how we should be thinking about prevention, about treatment.
Because if we're looking for the wrong thing...
Exactly. If we've been searching for these non-existent magic bullets against specialized weapons they don't have, well, that changes everything for the folks actually milking the cows.
Right. It directly impacts the bottom line. Let's talk about that impact. How much does this actually cost?
The numbers are pretty sobering. Estimates put the cost at around, $444 per clinical incident of E. coli mastitis, sometimes more.
Oof, per cow, per incident.
Per incident. And in many well-managed herds, E. coli can still be responsible for up to 80% of the really severe clinical cases.
80%, even in well-run operations.
Even then. So you're looking at major milk production losses, often persistent losses, plus treatment costs, the dumped milk, and sometimes the really hard decision to cull an otherwise good cow.
It all adds up incredibly quickly.
It does. So if our basic understanding of the enemy is flawed, it stands to reason our control strategies might be missing the mark too.
Which means wasted effort, wasted resources, wasted money. We need to really understand who these bacterial squatters are to actually, get ahead of the game.
Precisely. Understanding the true nature of the enemy is step one for effective control.
So what has the science actually uncovered that's causing this bacterial identity crisis? What's the evidence?
The really compelling evidence comes from looking deep into their genetics. Recent studies have just highlighted the incredible genetic diversity among E. coli strains pulled from mastitis cases.
Diversity? So they aren't all clones of each other?
Far from it. Unlike true specialist pathogens, which usually have a pretty consistent genetic toolkit for causing their specific disease.
Right, like they all carry the same weapons manual.
Exactly. These mastitis-associated E. coli, they show remarkable genetic heterogeneity. They're surprisingly different from each other, genetically speaking.
Heterogeneous. Okay, so more like a pickup basketball team than a drilled army unit. Everyone's got slightly different moves.
That's a great way to put it.
Yeah.
And the research has flagged a few key things. First, there's no single consistent set of virulence genes that you can point to and say, that's what makes it a mastitis E. coli.
No smoking gun gene.
Nope. No universal marker found in all mastitis strains and missing in all the harmless ones. Second big point. Most of these mastitis E. coli actually belong to the really common genetic groups, phylogroups A and B1.
Which are?
The same groups you find in totally harmless E. coli just hanging out in cow manure in the bedding around the farm environment.
Wow.
Which strongly suggests they're being recruited from the cow surroundings. They are some specialized invader arriving on a, Viking longship.
So they're not an outside enemy force. They're more like locals who know how to cause trouble when opportunity arises. Like the neighborhood kids who figure out how to jimmy the lock on the cookie jar.
Huh. That's a pretty good analogy, actually. And the third key finding ties into that diversity. Even when you look at E. coli causing seemingly identical clinical cases of mastitis, their genetic blueprints can still be wildly different.
Really? Same symptoms, different underlying bacteria genetically?
Exactly. And as Dr. James points out in the research, this genetic diversity is likely the big reason why mastitis vaccines targeting specific bacterial bits often give such inconsistent results.
Oh, that makes sense.
If there's no single universal target, a vaccine aimed at one specific version might be totally ineffective against another one the cow encounters next week. Like using a snowshoe in July, as you said earlier.
Okay, so if they're not specialized attackers packing unique weapons, what does make them so darn good at causing mastitis? You said they're ecological opportunists with enhanced fitness. What does that mean in practical bacterial terms?
Right. So instead of specialized attack plans, they seem to possess certain traits that just give them a big leg up in the specific environment of the mammary gland.
Like having the right tools for that particular job site.
Precisely. And one of the most consistently identified advantages, a really key one, is their ability to grab iron.
Iron, the stuff that makes things rust. Why is that so important inside an udder?
Well, iron is absolutely fundamental for bacterial growth. Yeah. They need it to multiply. But the cow's immune system knows this.
Defense mechanism.
Exactly. During an infection, the cow actively tries to hide iron to limit its availability. It's called nutritional immunity.
Okay.
But milk happens to contain a decent amount of citrate. And certain E. coli have a really efficient system, the ferric citrate uptake system, or FEC-operon, that lets them grab onto that citrate.
And use it like a key.
Sort of. Use it like a shuttle to pull in the iron. They desperately need to grow rapidly in that environment. Dr. Chen called it bacterial gold.
Bacterial gold. I like that.
And the studies back this up. E. coli that lack a working FEC system, much less likely to cause mastitis. Those with a really good FEC system, they tend to thrive and cause problems.
So it's a crucial fitness advantage? Tailored for that milk environment, not necessarily a weapon aimed at the cow.
Exactly. It's like having the best metal detector on a beach, is full of buried treasure. It gives you a massive advantage over competitors who don't have one.
Makes total sense. But surely it can't just be about iron, right? These bugs seem pretty good at surviving in there.
Oh, you're absolutely right. Iron acquisition is a big piece, but it's not the whole puzzle. They have other tricks up their sleeve, other metabolic adaptations.
Like what?
Well, many are very efficient at fermenting lactose, the main sugar in milk. That gives them a quick energy source for rapid growth.
Makes sense. Fuel readily available.
Yep. Some can also use other things for food, like alternative nitrogen sources, deserine, uric acid, which might be around, gives them more options.
Resourceful little things.
They are. Plus, they often have the ability to grow even when oxygen levels are low, which can happen inside the inflamed mammary gland. These are all factors contributing to their ability to just multiply like crazy once they get in.
Okay, so they're good at finding food, good at multiplying, but what about the cow fighting back? Doesn't her immune system try to, kick them out?
It absolutely does. And the E. coli that succeed in causing mastitis also tend to have some basic defensive capabilities. Nothing too fancy, maybe, but effective.
Such as?
Many have specific structures on their surface, LPSO antigens. They give them some protection against the complement system, part of the cow's first-line immune defense.
Okay. A bit of armor?
A bit. Some also show a degree of resistance to being eaten by neutrophils, those white blood cells that rush to the scene.
So they can dodge the cops sometimes.
Yeah. Or at least survive the encounter better. And they seem generally quite adaptable to the changing, inflamed conditions inside the udder.
But these aren't unique superpowers necessarily.
Generally not. As Dr. Chen emphasizes, these are mostly survival mechanisms found in many types of E. coli. The really important point is that a lot of the tissue damage we actually see in a case of mastitis.
The swelling, the bad milk.
Right, that's primarily caused by the cow's own massive inflammatory response to the rapid bacterial multiplication. It's the body's reaction, sometimes an overreaction, that causes a huge part of the problem.
Wow. So the bacteria multiply, and the cow's response trying to stop them actually causes most of the clinical signs we associate with the disease.
In many cases, yes. Especially the severe ones.
So this whole shift in understanding, MPEC to MAEC, opportunist, not specialist. It sounds like it has some pretty profound implications for how we manage mastitis day to day on the farm.
Profound is the right word. If we're dealing with adaptable survivors from the environment rather than targeted assassins, our whole prevention strategy needs, a serious rethink, or at least a refocusing.
How so? What changes?
Well, if these are environmental opportunists that just need the right conditions and fitness traits to thrive, then keeping them out of the udder in the first place becomes even more critical than we already thought.
It's like flood prevention, you said. Better to build strong levees than constantly mop up.
Exactly. It puts the emphasis squarely back on the environment.
So we're talking back to basics, but maybe with a renewed intensity. Clean, dry bedding is even more non-negotiable.
Absolutely paramount. Because we need to minimize that overall E. coli load in the cow's surroundings.
Yeah.
Remember, many of those environmental strains have the potential to cause trouble if they get the chance.
And this probably shines an even brighter spotlight on those high-risk times, right? Like transition, early lactation.
Definitely. Periods when the cow's own defenses might be naturally lower or teat ends might be more compromised. Managing those periods well becomes even more vital.
And what about the teat itself? Teat end health.
Moves right up the priority list. Healthy, intact teat ends and properly closing sphincters are the cow's primary physical barrier. That hasn't changed.
Still got to lock the door.
Still got to lock the door. And post-milking teat disinfection, even though these aren't typically spreading directly cow to cow like, say, Staph aureus,
it's still important.
Still crucial for reducing the number of bacteria hanging around on the teat skin, waiting for a chance to colonize the teat canal after milking.
Okay. And things like reducing teat end hyperkeratosis. Those rough ends.
Even more important now, perhaps. Those rough spots can be perfect little hiding places, little reservoirs for bacteria right where you don't want them.
And it also sounds like the cow herself, her own resilience, plays maybe a bigger role than we credited before.
I think that's fair to say. If it's about bacteria seizing an opportunity, then a cow with a robust, efficient immune system is much better equipped to prevent that initial colonization or clear it quickly if it does happen.
So maybe we need to double down on nutritional strategies that specifically support immunity during those high-stress times.
That makes a lot of sense in this context, supporting the cow's innate ability to fight back.
And could this even influence breeding decisions long-term, selecting for animals with genetically better mastitis resistance?
It absolutely strengthens the case for including mastitis resistance or indicators of immune competence in our breeding indices. All these host factors become even more relevant when facing an opportunistic threat.
Okay, so prevention gets a rethink. What about treatment if an infection does happen?
Yeah, this paradigm shift has significant implications there too, especially around antibiotics.
The antibiotic question. E. coli mastitis is kind of known for sometimes having high rates of spontaneous cure, right? Where the cow clears it on her own.
It is. Some studies show quite high self-cure rates, especially for milder cases.
Does this MAEC understanding kind of bolster the argument for maybe not automatically reaching for antibiotics in every single case?
It certainly adds weight to that discussion. Given that these might not be hyper-virulent bacteria designed to fight antibiotics, and the cow's immune system is often capable of clearing them.
Plus the whole antibiotic resistance concern.
Exactly. That huge elephant in the room. It pushes us towards a more thoughtful, more targeted approach. And because, as we discussed, the genetic makeup and therefore the antibiotic resistance patterns can vary so much.
From farm to farm, region to region.
Right. Relying on culture results becomes absolutely essential. Working closely with your vet to figure out if an antibiotic is needed, and if so, which one is most likely to work against the specific bug causing the problem on your farm.
So no more blanket protocols. It's about knowing your specific tenant and choosing the right eviction notice, or maybe no notice at all sometimes.
That's a good way to put it.
Yeah.
It demands a more individualized approach.
What about just focusing on the cow then? If a lot of the damage is from the inflammation.
Then focusing on managing that inflammation makes a lot of sense.
Yeah.
Could anti-inflammatory treatments like NSAIDs play a bigger, more central role? The evidence is growing in that area.
To reduce the damage caused by the cow's own response.
Precisely. And basic supportive care fluids for hydration, pain relief, especially in severe cases, that remains critically important to help the cow fight it off herself.
So it's really about having those detailed conversations with the vet. When is intervention beyond supportive care truly needed?
Exactly. And our sources even touch on some other interesting, maybe less conventional ideas.
Oh, yeah.
Like the fact that the number of viable E. coli bacteria naturally decreases in mastitis milk that's stored for a short time, and also that milk naturally acidifies, and that lower pH can actually inhibit or kill the bacteria.
Interesting. So time and acidity could be factors.
Potentially minor factors, but things worth discussing with your vet. Could these natural processes play any complementary role in an overall management strategy? Probably depends on the specific situation.
Okay, let's circle back to that iron connection you mentioned. The bacterial gold. That sounded really intriguing.
It is potentially a game changer.
If their ability to snatch iron using that FEC system is such a key advantage, could we target that, like put a lock on the gold vault?
That's exactly the line of thinking researchers are pursuing now. Dr. Martinez specifically highlights the FEC system as a potential Achilles heel.
An Achilles heel. Nice.
The idea is, if we could figure out ways to limit iron availability specifically to the bacteria without messing up the cow's own iron needs,
That would be huge.
it could be a major breakthrough in prevention or even treatment.
So what kinds of, futuristic approaches are being looked at? Are we talking iron blockers?
It does sound a bit sci-fi, but yes.
Yeah.
Researchers are actively investigating things like special iron-binding compounds.
Like chelators.
Exactly. Things that could potentially be given to cows during high-risk periods to basically scavenge the available iron before the bacteria can get it.
Wow.
There's also research into competitive inhibitors molecules that would physically block the bacteria from using their FEC system to grab the iron.
Come up the works.
Pretty much. And another angle is vaccines targeting the FECA receptor itself. That's the docking station on the bacteria where the iron shuttle binds.
So immunize the cow against the bacteria's iron door handle.
Essentially, yes.
Yeah.
Train the immune system to block that specific pathway. And even nutritional strategies are being explored, ways to maybe subtly adjust iron or citrate levels in milk to make it less hospitable.
But this is all still experimental stuff.
Oh, definitely. As Dr. Martinez cautioned, these are still in the research and development phases. But the key point is this deeper understanding of how these bacteria thrive, focusing on fitness traits like iron uptake, is opening up these completely new and potentially very exciting avenues for control.
Very cool. Okay, what about vaccination as it stands now? We've had E. coli mastitis vaccines for a while, like J5 vaccines.
Poor antigen vaccines.
But the results always seem a bit, mixed. Sometimes they seem to help, sometimes not so much. Does this new MAEC versus MPEC understanding explain that variability?
It absolutely helps explain. Remember that genetic diversity we talked about?
Yeah, the pickup basketball team.
Exactly. If you're dealing with this constantly shifting population of genetically diverse bacteria, the MAEC ecotype, rather than a uniform enemy, the MPEC pathotype, it makes sense why a single vaccine might not give consistent protection across all farms or all situations.
So how do the current vaccines like J5 actually work then?
They primarily work by stimulating the cow's immune system to produce antibodies against the core structure of LPS or endotoxin. This core structure is relatively conserved, meaning it's similar across many different gram-negative bacteria, including various E. coli strains.
Okay, so it's targeting a common piece, not the variable outer bits.
Correct. As Dr. James explains it, these vaccines might not reliably prevent the initial infection, and MAEC might still get in. But by having those antibodies against the core LPS ready to go, the cow's immune system might be able to react faster and more effectively once infection occurs. This could lead to quicker neutralization of the endotoxin that causes a lot of the systemic signs of illness.
Like the fever and shock.
Exactly. Or it might lead to more efficient clearance of the bacteria themselves by the immune cells. So the main benefit is often seen as reducing the severity of the disease rather than preventing it altogether.
So they're less like an impenetrable shield at the gate and more like giving the internal security force better training and equipment for when the gate crashers inevitably show up.
That's a really good way to frame it. It's about enhancing the speed and efficiency of the response.
And what's next in vaccine research, building on this MAEC idea?
Well, the research is definitely moving in new directions. As we just discussed, targeting those key fitness factors like the FEC iron uptake system components is one promising avenue. The hope is that could offer broader protection across more MAEC strains.
Makes sense. Target something they all need.
Right. There's also growing interest in finding ways to specifically boost what's called type 3 immunity in the udder.
Type 3 immunity.
Yeah, it involves specific immune cells called Th17 cells and signaling molecules like IL-17. This type of immunity is really crucial for recruiting those first responder neutrophils effectively and quickly to the site of infection.
So getting the army to the battle faster.
Exactly. And local immunization strategies are also being explored, ways to deliver a vaccine directly to the mammary gland tissue to try and trigger a stronger, more localized, and potentially more relevant immune response right where it's needed.
Interesting. So bypassing the systemic route.
Potentially. Yeah. As Dr. James pointed out, the overall focus is shifting. It's less about finding that one variable surface antigen to target and more about developing vaccines that fundamentally enhance the speed and effectiveness of a cow's initial immune reaction against a broader range of these opportunistic invaders.
Okay. Now, we can't talk bacteria these days without touching on antimicrobial resistance. Big topic.
Yeah.
Where do these mastitis-E. coli fit in? Are they becoming superbugs?
Well, the resistance patterns we see in mastitis-E. coli are complicated and highly variable.
Like everything else with these bugs.
Pretty much. The key finding seems to be that resistance isn't necessarily an inherent trait tied to the strains that are better at causing mastitis.
Oh.
Instead, the resistance profiles seem to be much more closely linked to the local antibiotic usage patterns on specific farms in specific regions.
So it reflects the drugs being used in that area?
Largely, yes. As Dr. Ken puts it, the resistance patterns are essentially a mirror reflecting local antimicrobial use, which, again, really hammers home the importance of doing culture and sensitivity testing.
To know what you're actually up against on your farm.
Exactly. You can't assume resistance patterns based on national or even regional averages anymore. It's about using antibiotics wisely, based on evidence, not just habit.
So is the sky falling? Are they resistant to everything?
Not necessarily, thankfully. The sources indicate that resistance to some really critical antibiotics, like the third-generation cephalosporins, is still relatively low in most places, often below 3%.
Okay, that's somewhat reassuring.
It is. However, the prevalence of strains resistant to multi-drug resistance is definitely increasing. And how common that is varies a lot by location.
And are there specific types of resistance emerging that are particularly worrying?
Yes, the emergence of strains producing extended spectrum bilectemases, or ESBLs, is a significant concern globally. These enzymes break down many common and important antibiotics,
making treatment much harder.
Much harder. And Dr. Chen also made another really crucial point. The environment inside the udder, in the milk itself, can actually change how well an antibiotic works.
How so?
Well, factors like pH, milk components, even the presence of immune cells can sometimes reduce the effectiveness of an antibiotic compared to how it performs in a simple lab test.
So what works in the petri dish might not work as well in the cow.
It's a possibility that needs to be considered. His advice was crystal clear. You absolutely need to work closely with your veterinarian to develop treatment protocols tailored to your specific farm, your specific situation, because those generic one-size-fits-all recommendations, they're becoming increasingly outdated and unreliable.
Makes sense. This really feels like it demands a constant evolution in how we manage mastitis, based on this better understanding. If we're mainly fighting opportunistic environmental bugs, what are the big frontiers for innovation? What's coming next?
One really exciting area is what you might call predictive microbiology.
Predictive microbiology sounds fancy.
It's the idea of developing tools that can help us identify, before they cause disease, which E. coli strains hanging out in the environment pose the highest risk.
How would you do that?
By looking for those key fitness traits we've been talking about. Like, does this strain have a really efficient FEC iron uptake system? Does it grow well on lactose? Dr. Olson actually envisioned a future where you might be able to, say, test your bedding.
Test the bedding.
Yeah. Test the bedding or manure slurry to identify if high-risk E. coli strains, those with the right fitness traits, are present at high levels. And if they are, you could implement specific targeted interventions before they get a chance to infect cows.
Wow, that would be moving way upstream from reactive treatment to truly proactive risk management.
Exactly, that's the goal.
What other big ideas are out there?
Well, given how tied these bacteria are to the environment, the whole concept of microbiome management is gaining traction.
Managing the microbes on the farm or in the cow.
Both, potentially. It could involve things like introducing beneficial bacteria probiotics into the environment, or maybe even the teat canal to try and outcompete the potential mastitis causers.
Competitive exclusion.
Right. Or maybe manipulating the environment, like bedding pH or moisture, to specifically create conditions that are unfavorable for bacteria possessing those key mastitis-associated fitness traits.
Making life harder for the bad guys.
Essentially. And maybe even exploring things like bacteriophages, those viruses that only infect bacteria, to specifically target and reduce populations of high-risk E. coli strains in the environment.
Using viruses to fight bacteria? Fascinating.
It is. And then, of course, there's continued focus on the host, the cow herself.
Right, strengthening the defenses.
Yes. Developing more targeted immunomodulators, things that could help the cow clear infections more efficiently, while, crucially, limiting that damaging excessive inflammation we talked about.
Fine-tuning the immune response.
Exactly. Plus, continued genetic selection for cows that just naturally have a more robust, efficient, and balanced immune response. And refining nutritional strategies to support that optimal immune function, especially during those critical high-risk periods like transition.
It really feels like a multi-pronged attack is needed. Environment, bacteria, cow immunity.
It has to be. Given the nature of MAEC as opportunists, there's unlikely to be a single silver bullet.
But even with all this new understanding, it feels like there are still some big question marks, right? Things we just don't know yet.
Oh, absolutely. Huge questions remain. For instance, we still don't fully understand why some E. coli infections flare up into severe, life-threatening cases, while others stay mild or clear quickly. Are there still some subtle, undiscovered genetic factors, maybe acting in combination, that contribute to higher virulence in certain strains?
That seems plausible. What else?
We need a much better handle on how long E. coli can actually persist in the teat canal without causing a full-blown infection. Days, weeks. And could we potentially manage the microbiome within the teat canal itself to prevent colonization by these MAEC strains?
The teat canal microbiome, that's a whole new frontier.
It really is. And how do other bacteria in the farm environment influence which E. coli strains become dominant and end up causing mastitis? There's likely a complex interplay happening there that we don't fully grasp.
The whole farm ecosystem.
Exactly. And practically speaking, developing rapid, reliable, maybe even cow-side tests that could help predict which infections are likely to become severe would be incredibly valuable. That could allow for much more targeted and timely intervention, focusing resources on the cows that truly need it.
Yeah, triaging cases better.
Right. And finally, really nailing down the precise transmission dynamics. How exactly do specific strains move from, say, fecal shedding into the environment and then ultimately into the udder to cause an outbreak? Understanding those pathways better could help us refine biosecurity and hygiene protocols even further.
Lots still to figure out.
Definitely. As Dr. Martinez put it, answering these questions is the next crucial stage of research. Getting those answers will hopefully lead to even more effective, more sustainable control strategies down the road.
Okay, so boiling this all down, for the dairy farmers, the vets, the industry folks listening right now, what are the main practical takeaways? What should they be thinking about or doing differently now based on this evolving picture?
There are definitely some key actions to consider right away. First, and maybe most importantly, double down. Seriously double down on environmental management.
Back to basics, but harder.
Yes. Clean, dry bedding isn't just good practice. It's probably your single most powerful weapon against these opportunists. Pay meticulous attention to high-traffic areas, freestall hygiene, moisture levels. Consider bedding treatments if appropriate for your system. Reducing the overall environmental load is critical.
Okay, number 1, environment. What else?
Second, schedule a dedicated conversation with your veterinarian, specifically to review your current mastitis treatment protocols in light of this MAEC concept.
Talk it through.
Yes. Does your current approach align with this understanding? Discuss the pros and cons of implementing on-farm culturing to guide antibiotic decisions. Talk about the potential role and appropriate use of anti-inflammatory treatments. Get on the same page.
Got it. Review protocols with the vet. Good.
Third, take a critical look at factors influencing cow resilience on your farm. How solid is your transition cow management? Are your nutritional strategies truly optimized to support mammary gland health and immunity during those high-risk times? And think about the long game. Are you incorporating mastitis resistance or related health traits into your genetic selection goals?
Boost the cow's ability to fight back. Makes sense.
And finally, number 4, keep your eyes and ears open for future innovations. Stay informed about the research into things like targeting iron uptake or new vaccine strategies. Be open to considering participation in research trials if opportunities arise. Maintain that dialogue with your vet, your nutritionist, your advisors about these evolving approaches.
Be prepared for what's coming next.
Exactly. This field is moving fast.
So it really sounds like the core message is shifting. It's less about trying to completely eradicate one specific enemy and more about creating an environment both outside and inside the cow where this diverse group of opportunistic bacteria simply has fewer chances to gain a foothold and thrive, while also maximizing the cow's own natural defenses.
That sums it up perfectly. And the expert voices in our sources really echo that. Dr. Olson emphasized that understanding these are common environmental bacteria helps explain why even the best farms have cases. Dr. Chins highlighted the need to shift vaccination towards boosting the early immune response because the bugs are so diverse.
Right.
Dr. Chen zeroed in on that shared need for iron, the bacterial gold, as a potential common vulnerability to target. And Dr. Martinez really drove home the opportunity for ecological control, for making the udder environment less welcoming to bacteria equipped with these specific fitness traits.
So the bottom line for everyone listening today is this. Our understanding of what causes E. coli mastitis has fundamentally shifted. We're moving away from thinking about specialized pathogens, MPEC,
towards understanding them as highly adaptable environmental opportunists, MAEC, equipped with fitness advantages for the udder environment.
And this new perspective has real practical implications for prevention, for treatment, for overall control strategies. The bacteria haven't changed overnight, but our map of how they operate has become much clearer.
A clearer map gives us a better chance of navigating the challenge.
Exactly. It opens up new possibilities for developing more effective, more targeted, and hopefully more sustainable ways to manage this incredibly costly disease. So, the final thought for you listening is, how will this new understanding inform your future mastitis management decisions? It might be time to take a fresh look at your protocols and see where adjustments could lead to better health and better productivity for your herd.
If there's one takeaway today, it's this. E. coli mastitis isn't about playing whack-a-mole with pathogens, it's about outsmarting an ecological opportunist. Ditch the magic bullet mindset and start asking, is my farm creating a 5-star resort for these bacterial freeloaders? For links to the full article, visit thebullvine.com. Hit subscribe next week. We're exposing why low SCC herds are secretly hemorrhaging profits. Until then, keep milking smarter, not harder.